choose the botanical fungicide because we want to change synthetic fungicides with the botanical fungicide with the safety and environmental effect. So in this lecture, Professor Tom will mention interesting topic. The next future Unpad Q collaboration in Atrapoda chemistry. Tom try to speak in try to write in bahasa. Unpad Q kolaborasi masa depan in Atrapoda chemistry. Big close from Tom. In his slide also combine in bahasa and English. He try to speak bahasa Indonesia very well.
Yeah.
Okay, then.
Okay.
Sound is yours.
Okay. There we go. Terima kasih banyak untuk datang di sini. Terima kasih banyak atas undangan yang diberikan kepada saya untuk ketiga kalinya mengunjungi Universitas Padjadjaran. Okay, jadi untuk ini lecture, bahasa Indonesia saya sudah banyak lupa, jadi mungkin lebih baik saya akan berbicara di bahasa Inggris, atau mungkin sedikit bahasa Indonesia, mungkin gado-gado. Alright, okay, so akan saya. Tidak kerja.
Yang dibuka itu.
Okay, so ini institute saya, nama saya Kebun Raya Botanic Q, Royal Botanic Gardens Q. I'm sorry. Hello.
Professor Tati.
Yeah.
The head of the postgraduate program.
Yes.
Very important person. All is the
student. Oh, I was okay. So I shall just saya sudah berbicara, saya akan berbicara mungkin bahasa Inggris saja, saya tidak tahu. So this is my institute, Royal Botanic Gardens Q. Buckingham Palace. So jadi mungkin 10 kilometer saja, so tidak jauh. So ini institute mungkin like 250 years old. Yeah. So, but I think Kebun Raya Bogor is actually older. Yeah, I think Kebun Raya. I'm not correct. I'm not 100%. Tetapi saya pikir Kebun Raya Bogor is the oldest botanical garden. Mungkin saya tidak tahu, tetapi mungkin. But yeah, so we're about 250 years old, and so we're not far from the center of London. And we have about, 2 square kilometers, 3 square kilometers. So we are famous for having the biggest collection of plants in the world, many plants from Indonesia. Many specimens. collected over 200 years of traveling around the world. Many fungal specimens as well. We have a big collection of fungal specimens. Mungkin juta spesimen jamur, mungkin collection besar di semua di dunia, tidak tahu mungkin. Ini saya di television CNN. Yeah. So also we have the biggest collection of seeds. Yeah, mungkin 2-3 juta. Mungkin banyak di Indonesia. Saya tidak tahu. So this is my research group. I have a new PhD student called Josh. This is PhD student Adam. He's working with, we're working on AI, machine learning, prediction of natural product mechanisms. With University of King's College London and University of Cambridge, UK. This is Niwayan. With the very famous Supratman. So hopefully we're going to do some interesting research on Aglaya. So I think everybody knows Aglaya is very interesting for cancer. So it could be very interesting. And then I'm going to have a new postdoc starting in 2025 for fungal natural products. So I don't know, tidak tahu belum. Mungkin tiga hari.
3 bulan.
3 tahun, yeah. Okay. So, okay, mungkin lebih baik saya akan berbicara Inggris. Okay. Yeah. So, the question for this lecture is, we're going to be talking about fungicides in Indonesia. Yeah. So we want to maybe do some research on the potential of plants to be fungicides in Indonesia. So first of all, we have to say, what is the problem with fungicides in Indonesia? Well, I can give you 3 examples. Methyl bromide is used in Indonesia, but menyerang lapisan ozone. So there's a problem with this one. Chlorothalonil is very, very popular fungicide in Indonesia, but padakun bagi ikan. Yeah. Propilet is also very popular in Indonesia, tetapi mahal sekali. So tidak bagus kamu membuat farming agriculture di Indonesia dan ini banyak wine, ya? Okay. Another problem. Ada masalah di triazole fungicide. Kamu tahu triazole? Ya. Okay, pertanyaan, apa masalah dengan triazole? Tahu? Bukan? Yeah. Jadi, okay. So the problem with triazole is in agriculture, we are using this triazoles, yeah? Similar. And in rumah sakit, also triazoles for fungal infection in humans. Yeah. So if we use this triazole in Indonesian agriculture, the problem is that fungal infections for humans become antibiotic resistant. How would you say this? How would you explain this? So basically, we're using the same or similar compound in agriculture and similar compound in hospitals. So then we have natural selection, and fungal pathogens become resistant. So we're seeing Aspergillus fumigatus is a fungal species in agriculture, but also infects humans. So the problem is that basically, if we keep on using triazoles, then we can't use triazoles both for agriculture and in the hospital. It's not tidak bagus, yeah. Okay. So the specific problem is, let me see if I can translate this. How are we going to say that the fungus, the fungal pathogen in a hospital is adapting, is learning how to, yeah, it's adapting to the triazole, becoming resistant. Yeah, so it's becoming azole resistant. Yeah. Okay. So, yeah, so this is one big problem. Yeah. So what is the potential solution to this problem? So the problem is this one. Maybe the solution is fungisida alami yang terbuat dari tumbuhan telah menjadi alternatif yang popular. Yeah. So here we have some examples. 1, 2, 3, 4. Impact example of natural fungicides. So you can see these products are already available on the market in, it's already used in Europe and in North America. So what compound is present? So this one, we see transneurodiol. Yeah. This one here is terpium for monoterpene. And this one, fracture, is a peptide. Yeah. So this is the existing example in the world of a natural botanical fungicide. So one question, pertanyaan, okay, dari Indonesia ada banyak pohon, ya. Jadi mengapa tidak ada fungicide natural dari Indonesia? Belum. Okay. So, yeah, ini transneurodiol terpium for pertanyaan apa kerja ini, ya? Dua senyawa bekerja pada fosfolipid bilayer, ya. Kamu semua tahu fosfolipid bilayer? Ya. Jadi dengan oh tidak kerja. Okay, jangan kutip. Um, no. It's okay. Yeah, it's just the pointer doesn't work.
Oh, yeah.
Okay. Jangan kutip. So these compounds are disrupting the phospholipid bilayer, and then the fungus is dying. Yeah. So when the phospholipid bilayer is broken, the fungus dead. Yeah. So how about if we want to ask the question, how can we know if the phospholipid bilayer is broken? Unain, how do we say phospholipid bilayer is broken in Indonesia?
Jadi merusak lapisan fosfolipid dan dua lapisan fosfolipid.
Yeah. Okay. So we can do this. We can use propidium iodide. Okay, so misalnya propidium iodide tidak bisa thank you. Okay, so ini propidium iodide. Okay. Yeah, ini propidium iodide. So propidium iodide, kamu tahu propidium iodide, bukan? Okay, so then propidium iodide, kapan propidium iodide masuk di sel, propidium iodide fluorescent, ya. Tetapi kapan propidium iodide bukan masuk di sel, bukan fluorescent. Ya. Jadi kalau apa ini phospholipid bilayer sudah apa? Rusak. Okay. Propidium iodide bisa di sini propidium iodide kerja fluorescent, ya. So lihat ini saya kerja ini sudah dengan SDS. Okay, kamu tahu SDS? Sodium dodecylsulfate. Yeah. So sodium dodecylsulfate is a membrane disruptor, phospholipid bilayer disruptor. So saya sudah membuat ini. Okay, ini untuk 20 minit, 50 minit. Okay, ini control. Okay, ini fluorescence, ya. Dengan plate reader. The phospholipid bilayer. Oh, but now, yeah. Okay. So, wait. So ini example dan ini example kerja di phospholipid bilayer. Okay. Tetapi ini example, okay, bukan phospholipid bilayer. Ini peptide kerja di how do you say iron?
Iron, same.
Same. Okay, sama iron. So ini peptide kerja di iron chelator. That means like how would you say iron chelator? It means it takes the iron. Yeah. Okay. Jadi, okay. So saya kerja satu publication dengan ini fungicide nama fracture, ya. Jadi kami sudah membuat transcriptomics. Okay, karena kami tidak tahu ini ini fungicide fracture tidak tahu berapa kerja, ya. Jadi kami sudah membuat transcriptomics dan kami sudah lihat ada banyak genes genes functional to iron, ya. Jadi kami pikir mungkin ini fracture kerja di iron chelator, ya. So kami sudah membuat eksperimen dengan supplementation with iron, and this sudah tidak kerja dengan ada banyak iron. So kami sudah pikir fracture ini peptide kerja di iron saja. Okay, so pertanyaan, bisakah kita menemukan fungisida nabati dari tanaman Indonesia untuk melindungi tanaman cabai? Cabai. Maafkan saya. Ya. Yeah. Okay. So di sini mungkin ini this is our project proposal. Yeah. So step one, we want to collect 5,000 Indonesian plants from Kebun Raya Botanic Garden. Yeah, Kebun Raya Bogor. Okay, 5,000 besar sedikit, ya. Tetapi mungkin dengan impact student, mungkin satu student bisa membuat collection mungkin 50 sampel setiap hari. Okay, mungkin dengan impact student, mungkin dengan impact bulan kami bisa membuat 5,000, ya. 5 juta, ya. 5,000, yeah. Okay, and then extract the sample, and we put them into 96 well plates. So this we call like an extract library, and then screening the sample with fungal pathogen. So I think Dr. Enda Julia can do this experiment. Isolation of the active compounds, Professor Unai, with some students. And then more testing with fungal pathogens. And then at Q, we can do toxicology of the active compound. And then in UNPAD, we can do testing on plants. And then in we can also do mechanism of action, which is important to understand the mechanism of action. Yeah, so 4 students, 25 samples, 3 bulan, mungkin, ya, kami bisa kerja ini.
Recording stopped.
Simple protocol, but we can get results in plants. Okay. So now we have from 5,000 plants, we have active compounds that works in plant infection with fungus. Oh, I can catch it. Okay. So another question is the compound toxic? So I have a collaborator, we can do predictive toxicology. So a new idea for toxicology, like a new idea is we can make a prediction using artificial intelligence. So the computer can understand, based on the structure, does this compound look similar to another compound that is a problem? So the computer can make some kind of prediction about toxicity. Yeah, actually, one more thing I would say about this. So this is like a big idea at the moment is in silico pharmacology. But the only problem is it's based a lot on synthetic compounds. So if you have a natural compound, maybe the computer doesn't understand because it can't find a similar compound. So if you're testing synthetic compounds, the computer can make a comparison with other compounds. If you have a novel plant compound from Indonesia, maybe the computer does not understand what this compound is. That's the only problem, I think. Uh-oh. Yeah. Okay. So last question is we want to know, how does the fungicide work? How does the chemical kill fungi? Why do we want to know this? Because if you want your fungicide to be accepted in Indonesia, probably the government wants to know, how does this fungicide work at the molecular level? And if you want to have good publication, it's important as well. So this is what one of my areas of interest. So I do this kind of work a little bit complicated to explain. What we do is we take the compound and we give it to
you